My name is Steve Strauss. I'm a professor in the Department of Forest Ecosystems and Society at OSU and director of the OSU Outreach and Biotechnology program. It is my pleasure to welcome you to this lecture tonight. This is the last of 5 lectures in what is now the 7th year of the Food for Thought lecture series, sponsored by the OSU College of Agricultural Sciences and Forestry. Its mission is to bring leading scientists and scholars to speak directly to the public on urgent issues of biotechnology in relation to food production and sustainability. Unfortunately, due to budget constraints, we are still not sure if the lecture series will continue after this year. However, if this is the last Food for Thought event, I can think of no better way than to end and no better speaker than to end on. Genetically engineered, or GE, crops are grown over huge acreages in a number of countries, including the USA, Brazil, Argentina, China, India, and Canada, and about a dozen others. They are intrinsic parts of agriculture and foods in these places. 2 types of GE traits, herbicide and insect resistance, account for the large majority of the acres that have been sown. Both types have had major and complex environmental and economic impacts, some really good, some not so good, some planned and some unplanned. Understanding these impacts and putting them in a proper context is not a simple task. Tonight's speaker, Yves Carrière, has produced some of the highest quality and most highly cited ecological studies of GE crops. He is supremely qualified to give us a scientifically accurate, integrated, and unbiased view of their ecological effects. Yves was educated in Canada, obtaining his bachelor's at Laval University in Quebec and his PhD at Simon Fraser in British Columbia. He worked as a postdoctoral researcher and then an adjunct professor at McGill and Laval for a number of years afterwards. He joined the faculty of the University of Arizona in 1998, where he has remained to this day and is currently a professor in the Department of Entomology. Yves' scientific output has been prodigious. He's published more than 140 scientific papers, the large majority of which have been in top-tier peer-reviewed journals. He is also widely recognized as a source of high-quality advice by peers and government boards. He serves on editorial boards of 2 journals and has served on a dozen different advisory panels in the US and abroad. He's obtained awards for teaching from the University of Arizona and for research from the Entomological Society of America. When the US government needs advice on complex matters of science policy, it often asks the National Academy of Sciences to assemble a team of leading scholars to provide a state-of-the-art consensus report. Yves served on such a panel in 2009 through 2010. Its charge was to review the impact of GE crops on farm economics and sustainability in the USA. Tonight, Yves will summarize and update the ecological findings of this report and provide his own views of the impacts of GE crops in the US and around the world. So please help me in welcoming Yves to Oregon State University.
So I'd like to thank Steve for the wonderful introduction and the invitation to give this talk. It's a pleasure for me to be here tonight and to talk about the environmental impacts of genetically engineered crops. So most of what I will talk about tonight, as Steve said, is based on an NRC report that was published at the end of the summer of 2010. And I would like to take a minute to acknowledge the work of the people that have worked to write this report. So since this report was published, there has been a few changes with respect to the environmental impacts of GE crops, but most of these changes have been quantitative. rather than qualitative. And for that reason, most of what I will discuss tonight is based on this report, although I will talk about new data. So the first thing that we have to do here is define what is a genetically engineered crop. And it's simply a plant that contains genes from different species, which were inserted in the plant's genome by genetic engineering techniques. So for example, here you have a cotton plant with its own genome. And here you have a soil bacterium called Bacillus thuringiensis, which happens to produce proteins that are toxic to some insect species. So what you do here is you take this gene and insert it in the cotton genome such that now you have a GE crop that will produce one Bt toxin that will confer protection against some insect pests. So far, the first generation of GE crops in the United States have been engineered for traits that confer greater protection against pests. So the first category of these crops are crops resistant to herbicides such as glyphosate and glyphosate. And the crops that are commercialized in the United States right now are alfalfa, canola, corn, cotton, soybeans, and sugar beet. Then you have crops protected against some key pests, crops that produce the toxin from Bacillus thuringiensis, and those are Bt corn and Bt cotton. And lastly, you have a rarer category of crops that have been engineered for resistance to plant viruses. These are virus-resistant papaya that are only used in Hawaii, and virus-resistant squash that are used in several states of the United States. So by far, most of the GE crops used in the US are soybeans, cotton, and corn that have been engineered either for resistance to herbicides or for the production of Bt toxins. All these crops, those three main crops, were commercialized in 1996, and their use has increased rapidly in the United States. So for example, in light blue here, you have herbicide-resistant soybeans. In 1996, you had about 10% of the soybeans in the US that were resistant to herbicides. And you can see that the use has increased rapidly, such that 94% of all soybeans grown in the United States were engineered for resistance to herbicides by 2011. So if you look at corn and cotton, you're looking at between 65% and 75% of all corn and cotton in 2011 that were either engineered for resistance to herbicides, production of Bt toxins, or both. The United States is the country in the world that uses the most transgenic crop, but those crops are used in 28 other countries. There are transgenic crops used on every continent but Antarctica. If you look in 2010, the global acreage of transgenic crops corresponded to about 10% of all rain-fed cropland worldwide. So clearly, farmers have adopted transgenic crops, and it's likely that more transgenic crops will be used in agriculture in the future. So even though farmers have adopted transgenic crops, there have been some concern generated by the use of these crops. Some of these concerns about potential negative environmental impacts of these crops. And for that reason, we will talk about the environmental impacts of genetically engineered crops, mainly in the United States tonight. As I said, using mainly data from the NRC report, but also using data published between 1996 and 2012. So what I'll do tonight is first start about the environmental impacts of herbicide-resistant crops. Then I'll talk about the impacts of insect-resistant crops, Bt crops. And lastly, I'll talk about environmental issues that are related to both crop types. And those are gene flow between GE crops and non-GE crops, and gene flow between GE crops and weeds. So let's start by herbicide-resistant crops. Herbicide-resistant crops are crops that can be sprayed with an herbicide and they will survive, but the weeds in the fields that are sprayed, most of them will die. So most crops in the United States have been engineered for resistance to glyphosate, and that's mainly what I will talk about tonight. And glyphosate is an herbicide that has relatively low environmental impacts compared to other herbicides. It has low environmental impacts because it has a low toxicity for animals, birds, and fish. And also it has a low soil and water contamination potential because it binds readily to soil particles, and also because it has a short half-life. So it is degraded readily by bacteria. So the first impact of herbicide-resistant crops is that they have favored a replacement of herbicides with greater environmental impacts by glyphosate that has lower environmental impacts. So here what you have are data for soybeans in the United States. On the y-axis, you have the pounds of active ingredients of herbicides used per acre of soybeans in the United States. And you can see in black here, starting in '96, that the use of glyphosate increases as the adoption of herbicide-resistant soybeans increases. At the same time, you can see that the use of other herbicides decreases. Now here, if you look at this red arrow, you have the global use of active ingredients of herbicides in 1996 in soybean, and then in 2007, the last year for which we had data, and you can see a slight increase in the use of herbicides globally in soybeans. So from this, we conclude that glyphosate has replaced other herbicides, and we see a trend for a slight increase in use of active ingredients of herbicides in soybeans. And the situation has been the same, more or less the same in cotton, so I will not present those data. In corn, we have a slightly different situation. So you can see the increase in use of glyphosate that parallels the adoption of herbicide-resistant corn. And you can see a decline in the use of other herbicides. But you can see that glyphosate has replaced the other herbicides to a lower extent. So if you look at the global use of herbicides, you can see that it has slightly decreased in corn from 1996 to 2007. The second and perhaps the most important environmental impact of herbicide-resistant crops is that it has contributed in reducing the use of tillage, which is used by farmers to prepare their soil before planting. So in the United States, you have roughly 3 types of tillage. You have conventional tillage, whereas all post-harvest residues are plowed into the soil before planting. So if you look at a field before planting, you essentially look at a field covered with bare ground. Then you have conservation tillage, when at least 30% of the soil surface is left covered with crop residues. And finally, you have no-till practices where crop residues are left undisturbed and the next crop is seeded directly into the soil. So for example, here you see soybeans that have been planted directly over an old field of wheat. So conservation tillage and no-till practices have several positive environmental impacts. They can reduce soil erosion. They can increase water infiltration in fields and reduce runoffs of sediments and chemicals. So they reduce agricultural pollution to surface water. They can improve soil quality and moisture retention, so they can increase the health of agricultural soils. And lastly, they can increase carbon sequestration and therefore mitigate the impacts of global warming. So what the data show is that the introduction of herbicide-resistant crops have contributed in reducing conservation and increasing conservation tillage and no-till practices in the case of soybeans and cotton, but not in corn. Top panel here, you have the acreage of soybeans managed by different tillage practices starting in 1990. And you can see in gray here an increase in the use of conservation tillage up to 1996, when the herbicide-resistant crops were commercialized, and an increase in no-till practices here. And after 1996, further increase in conservation tillage and further increase in no-till practices. In cotton, a similar situation, slight increase in conservation tillage, very, very slight increase in no-till practices. After 1996, greater increase in no-till practices, greater increase in conservation tillage. In corn, we don't see this trend. There's an increase, but it plateaus before 1996, and then it doesn't change. So here we're looking at temporal data, longitudinal data, and there may be many reasons as to why the tillage practices have changed, but it's important to say that other studies are showing a clear link between use of herbicide-resistant crops and reduction in tillage used by farmers. So here you have one such study. You have use of conservation tillage and no-till practices in farmers that use herbicide-resistant soybeans in 1997, as opposed to farmers that planted conventional varieties. And you can see significantly higher use of conservation tillage and no-till practices in farmers that use herbicide soybeans than in farmers that don't. So in summary, what have been the main environmental impacts of herbicide-resistant crops? They have favored the replacement of herbicides with negative environmental impacts by glyphosate, which has less environmental impacts. They have favored a reduction of tillage, which has positive environmental impacts. It can minimize soil erosion, increase soil quality and carbon sequestration, and improve quality of surface water. Finally, we have seen a global that the use of herbicide active ingredients has slightly decreased in corn, but slightly increased in soybeans and cotton. So most environmental impacts of herbicide-resistant crops have been positive, and there are two factors that could cut short those benefits. Those are resistance to glyphosate in weeds and weed shifts. So resistance to glyphosate in weeds is defined as a genetically based increase in the frequency of resistant individuals that occurs following exposure of a weed population to glyphosate. So what happens here is that when you use glyphosate, you will have a few individual weeds that will have resistance alleles, and these weeds will survive better than the weeds that don't have resistance alleles. So if you use glyphosate year after year in an agricultural field, you will select for individuals that bear resistance alleles. So the frequency of the resistance alleles will increase, so the frequency of resistant weeds would increase, and with time you will see that glyphosate will be less efficient to control the species of weed. Weed shifts occur, it's a slightly different phenomenon. It occurs when you observe a change in the local community of weeds that is favored by a change in the methods that are used to control weeds. So what happens here is that glyphosate is a broad spectrum herbicide, so it kills most weed species, but you have a few species that will be naturally tolerant to glyphosate. So if you apply glyphosate year after year in an agricultural field, the weeds that are naturally tolerant will survive better than the weeds that are not. And then you will progressively see a change in the weed community such that more and more species will be tolerant to glyphosate. So with time, glyphosate becomes less efficient at controlling the weed community in agricultural fields. So when herbicide-resistant crops were commercialized, there were no weeds resistant to glyphosate in the United States. And you can see the light bars that the number of species that have evolved resistance has increased, such that today in the US there are at least 10 species that have evolved resistance to glyphosate. The dark bar are the number of species that have evolved to glyphosate worldwide. So here you have two such species, two of the most common. You have horseweed, which has been a problem in herbicide-resistant corn, cotton, and soybean fields. This is a weed that has been, a resistant weed that has been found in at least 14 states of the US over more than 2 million acres of crops. So here you have soybean plants, and in the background you have horseweed growing. Another problematic weed has been Palmer amaranth. It has been a problem specifically in herbicide-resistant cotton and soybeans field. It has been noted in at least 5 states, over 200,000 acres of crops. And you can see here soybean plants, and between the soybean plants you can see Palmer amaranth growing. So clearly not a good situation for farmers that are trying to grow soybeans. Here you simply have a list of weeds that went through weed shifts that became more frequently encountered in fields of herbicide-resistant crops. So what have been the consequences of resistance and weed shifts? First, farmers have increased the frequency of use of glyphosate in their fields. They have started to use other herbicides again to control the weeds. And they have reverted to traditional methods for control weeds. That is, they have used more tillage to control the weeds. So the impact of resistance and weed shifts is that they have reduced the environmental benefits associated with herbicide-resistant crops. So what we have learned in the last 15 years is that more diversified weed management strategies and crop production practices are needed to mitigate the problem of resistance and of weed shifts. Now let's talk about the environmental impact of Bacillus thuringiensis crops, Bt corn and Bt cotton. So Bacillus thuringiensis, the soil bacterium, is a natural and widespread pathogen of some insects. So if you do some gardening, at the end it's likely that you will have Bt bacteria on your hands. We know that Bt is not toxic to people and to most non-target organisms. And we know that the use of Bt is much safer than the use of most conventional synthetic insecticides used in agricultural fields today. And we know this because Bt has been used as sprays to control forest pests and in organic agriculture for a very long time. And it has also been used as Bt granules for the control of some vectors, such as mosquitoes. So the first environmental impact of Bt crops is that they have contributed in reducing the use of synthetic insecticides in cotton and corn fields in the United States. So what you have here is the amount of pounds of active ingredients of synthetic insecticides used per acre of cotton in the United States from 1996 to 2007, the last year for which we had data when we wrote the report. If you look around 1996, you can see that about 2 to 3 pounds of synthetic insecticides were used. And as the use of Bt cotton increases in the United States, you see a decline in the use of synthetic insecticides in cotton. Here we have similar data for corn. 1996, you're looking at between 0.2 and 0.3 pounds of synthetic insecticides per acre of corn. So about 10 times less synthetic insecticides are used to grow corn than cotton in the United States. And you see a progressive decline in the use of insecticides as the adoption of Bt corn increases in the United States. So again, here we're looking at longitudinal data, so there may be many factors that may have contributed to the lower use of insecticides. But again, there are other studies that show a clear link between the use of Bt crops and the reduction in use of insecticides. For example, in Arizona, we have shown that there's about 30 to 60% less synthetic insecticides used in Bt cotton than in non-Bt cotton. And we have shown that by looking at the same region during the same year. So depending on the year, you use between 30% and 60% less synthetic insecticides in Bt cotton than non-Bt cotton to control insect pests. A second positive environmental impact of Bt crops is that their deployment has contributed in reducing the population of some target pests regionally. So what happens here is that when you use a lot of Bt crops in a region, the females of the target pest will lay many eggs on the Bt crops. And the Bt crops are very good at controlling those target pests, so the eggs will produce very few individuals. So if you have enough Bt crops in a region, it's possible that you will suppress the capacity of growth of the pests enough to observe a regional suppression of their pest populations. So 4 key pests of cotton, in 4 pests of cotton, we have seen such regional declines, 3 in the US, Eleuthis virescens, Helicoverpasea, and pink bollworm in Arizona, and 1 in Arizona, in China. And we have also seen regional declines in the populations of the European corn borer, a key pest of corn in the Midwest. So what I'm showing here are data that show regional decline in pest population of the European corn borer, which is a pest specifically targeted by Bt corn. So what you have here on the y-axis is the population density of the larvae of the European corn borer in fields of non-Bt corn. And you can see that starting in 1962 in Minnesota and Illinois and Wisconsin, you can see that the populations fluctuate. In some years, the larval population is quite high, and some years it's quite low. The shaded area corresponds to the start of the use of Bt corn in Minnesota. By 2010, about 60% of the corn planted in Minnesota produced Bt toxins. And you can see that as the use of Bt corn increases, the population density of the European corn borer declines both in non-Bt corn and in all fields of corn. And you see the same trend in Illinois and the same trend in Wisconsin. So in this study, it was shown that those regional declines have resulted in significant economic benefits, not only for farmers that plant Bt corn, but also for farmers that plant non-Bt corn. And as I said before, the use of Bt corn has reduced the use of synthetic insecticides, but it's likely that when those regional declines occur, you will use less insecticides in fields of the non-Bt crop, fields of non-Bt corn also. Another positive environmental impact is that Bt cotton has been used to eradicate a key pest of cotton in Arizona and the west of the United States. So that pest is the pink bollworm, which is a pest that has been introduced to the United States in 1925. And since its introduction, tons of insecticides have been used to control this pest in cotton. So what farmers have done in Arizona here is starting in 2006, they have planted close to 100% Bt cotton to eradicate this pest. Bt cotton kills more than 99% of the larvae of the pink bollworm. And at the same time, sterile insects were released to prevent the evolution of resistance. So what you have here is data on percentage infested bolls in fields of non-Bt cotton in Arizona between 1997 and 2005. And you can see that the percentage infested bolls vary between 10 and 30%. So we're looking at a key pest of cotton here. In 2006, 1/3 of the state was planted with close to 100% Bt cotton. 2007, 2/3. 2008, full state. By 2009, there were very few pink bollworm left. Here you have the number of sprays used per field per year to control pink bollworm. Between 1996 to 2005, you're looking at, on average, 1 spray per year. The thing to be said here is that from 1996 to 2005, farmers in Arizona have been using a lot of Bt cotton. And when you use Bt cotton, you don't need to use sprays to control the pink bollworm because Bt cotton kills virtually 100% of the larvae. So that explains why we're looking at only 1 spray. If we were looking at 94, 95 before Bt cotton, we would be looking at an average of at least 3 sprays just to control the pink bollworm. So what you can see here is the use of sprays in cotton has declined. In 2009, there was not a single spray used to control this pest in Arizona. Now, sterile insects, why did farmers release sterile insects, it's because if you grow 100% Bt cotton, you kill the susceptible insects, but there will be a few individuals with resistance alleles that will survive on Bt cotton. So sterile insects were released over cotton fields such that those sterile insects would mate with the rare resistant individuals that survive, and if that happens, the progeny is non-viable. So sterile insects were released to prevent the evolution of resistance during the eradication program. Now, Bt crops produce Bt toxin during the entire growing season. So even though we know that Bt toxins are specific, there were some concern that because the proteins are there for a long time, that they could have negative impacts on non-target organisms, insects that we don't want to kill. So there have been two types of studies that have been done to look at this. The first look at the impacts on biodiversity in general. The most common studies have looked at the impacts on organisms providing important ecological functions, such as natural enemies, pollinators, and soil organisms. So before showing some data on this, what I want to say here is that Bt proteins are specific in their mode and the way they affect pests. So here what you have are the first cultivars of Bt crops that were commercialized in the United States. Bt corn in 1996 was only producing a single toxin, which is called Cry1 Ab. Bt cotton, Cry1 Ac. Those toxins are very similar, and they all target lepidopteran pests. So here you have the mortality induced by those Bt toxins on different lepidopteran pests of corn and cotton. And you can see for European corn borer, Eleuthis virescens, pink bollworm, Bt cotton kills, or corn, kills virtually all larvae. So it works well. But if you look at Helicoverpa zea, armigera, which are key pests of corn and cotton, mortality starts to decline between 60–95%. And if you look at Spodoptera exigua, which is a pest that feeds on corn and cotton, less than 20% of the larvae will die. So what this shows is that the Bt proteins are specific. Even within the target group, the Lepidoptera, they don't necessarily kill the insects. So because of this, it was anticipated that most non-target organisms outside of the target group would not be impacted. So here's a study that we did in Arizona to look in general at the impact of Bt cotton on beetles. So the model system was beetles. And we use beetles because it's a highly diversified group that contains a lot of species. And those species have very different ecological roles. So you have beetles that feed on plants, beetles that feed on pollen, beetles that feed on other insects, beetles that are omnivores, that eat meat and plants, that are detritivores. So what we did here is we compared the biodiversity of the beetles across 81 cotton fields, some non-Bt cotton, non-GE, some Bt cotton engineered for resistance to herbicides. And we measured biodiversity as the density of beetles in cotton fields and the number of species. And here you have a summary of the outcome of this study. In blue, you have factors that had significant impact on the biodiversity of beetles. The use of synthetic insecticides has significant impact. The use of an insecticide, it's called an insect growth regulator to control the whiteflies, which is a key pest of cotton, had a significant impact. The density of cotton plants in a cotton field, again, a significant impact. And the diversity of the plants around the cotton fields had an impact on biodiversity. Here, we contrasted non-GE versus GE cotton, and it's in black because we found no difference in biodiversity between non-GE and GE fields. And here we contrast Bt versus Bt herbicide-resistant fields, and we find no significant difference. So the conclusion from this analysis is that there are a lot of factors that affect biodiversity of beetles in cotton fields, but whether the cotton is GE or not is not one of these factors. So here you have the other type of study that more directly looks at impacts on specific groups of non-target insects. And this is a meta-analysis that summarizes many field studies that were done to compare the density of the groups in plots of Bt cotton versus non-Bt cotton, or plots of Bt corn versus non-Bt corn. And none of those plots were sprayed with synthetic insecticides. So any difference in density that we find would be due to the presence of the Bt toxins. So the groups that we are looking at are predators, parasitoids, mixed group that have more than one function, herbivores, omnivores that feed on meat and plants, and detritivores. So on the top panel here, you have the results for the comparison between Bt and non-Bt cotton. The y-axis here is an estimate of the average size of Bt versus non-Bt cotton. So if a bar is positive, there's 0 here, it means that the density of a group will be higher in plots of Bt cotton than in plots of non-Bt cotton. So you can see that the bars are all negative. So there's a trend for finding a lower population density of all those groups in plots of Bt cotton versus non-Bt cotton. Now here you have what is called a 95% confidence interval. And if this overlaps 0, this means that the effect is not statistically significant. Or another way to say that is to say that the effect is small. So if you look across the groups, you only find a statistically significant effect for predators. The number here is the number of independent comparisons used to calculate those effects. And you can see here that we're summarizing many, many studies. So to come back to the significant effect of predators, it was driven by 2 groups, the Nabidaes and the Coccinellidaes. The Nabidaes are a family of predators that feed on lepidoptera. So the reason for their lower abundance is probably that there were less lepidopteran insects in Bt cotton than non-Bt cotton. So if there's less food, you find less predators. Here we have the Coccinellidae, which do not necessarily feed or rarely feed on lepidopteran insects. And we do not know why the Coccinellidae were rarer in plots of Bt cotton than in plots of non-Bt cotton. So more work will be needed to understand what's going on here. Here we're looking at the results for corn, and again, most of the differences are non-significant, except for the parasitoids. So if you look at the significant effect of for the parasitoids, in 93 of the 116 comparison, we were looking at 1 species, Macrocentris grandii. And that parasitoid is a specialist on European corn borer. So in plots of Bt corn, you don't find any European corn borer. So it's not surprising that this parasitoid was in lower density in plots of Bt corn than in plots of non-Bt corn, because there was no food for this parasitoid there. So what these studies suggest, therefore, is that Bt toxins in transgenic crops have very little impact on non-target organisms. So the same idea here, we have a meta-analysis that summarizes the results of lab studies on the effect of Bt proteins or Bt pollen on survival of honeybees. And here we're looking at 2 types of Bt proteins that target lepidopteran pests, proteins that target coleopteran pests. A positive value means that the survival of honeybees is better if they feed on Bt proteins or on Bt pollen than if they feed on a sugar solution or on non-Bt pollen. But you see here that the confidence interval overlaps 0, so we find no significant effect of those Bt proteins on survival of honeybee, which is an important pollinator of many crops. So if we summarize the main environmental impacts of Bt crops, we find that their deployment has contributed in reducing the use of synthetic insecticides in corn and cotton. We have observed regional declines in populations of some target pests. It has been used as a tool to eradicate an introduced pest of cotton in the western United States. And many studies suggest little or no adverse effects on non-target organisms. Again, as in herbicide-resistant crops, one factor could cut short those benefits, and it's the evolution of resistance to Bt in pests targeted by Bt crops. So what you have here is the number of pests that have evolved resistance to Bt crops worldwide. Between 1996 and 2002, no pest evolved resistance. 2003, first pest that evolved resistance is Helicoverpa that evolved resistance to Bt cotton in some states of the United States. And then you see more species evolve resistance, such as today, at least 8 species have evolved resistance to Bt plants. In the United States, we have 3 species that have evolved resistance. Zea evolved resistance to Bt cotton. The fall armyworm has evolved resistance to Bt corn in Puerto Rico. And the western corn rootworm has evolved resistance to Bt corn in some locations in the Midwest. So clearly, pests can evolve resistance to Bt crops, and for that reason, we need measures to delay the evolution of resistance to preserve the positive environmental benefits of Bt crops. And the measure that has been popularized to manage the evolution of resistance is called the refuge strategy, which is a strategy that has been mandated by the US EPA to delay the evolution of resistance in pests. So the way this works for a cultivar that produces a single Bt toxin is as follows. In blue here, you have what we call refuges, which are crops that do not produce a Bt toxin, or host plants used by the target pests that do not produce the Bt toxin. And those refuges will produce lots of target pests that are susceptible to the Bt toxins, so they don't have resistance alleles. The yellow field here is a field of a Bt crop, and an interesting phenomenon for resistance to Bt is that resistance is generally recessive, which means that for an individual to survive on a Bt crop, that individual needs to have 2 resistance alleles. So those individuals are generally rare because when we commercialize a Bt crop, the frequency of the resistance alleles are rare, which means that the individuals that have 2 such resistance alleles will be even rarer. So what happens here is that if you have a refuge that is close to a field of a Bt crop, those susceptible insects will mate with the rare resistant individuals. And the progeny will only bear 1 resistance allele, so the progeny will be killed by the Bt crop. So what happens here is that if we have refuges close to every field of a Bt crop, the resemblance between the resistant parents and their offspring is close to 0, which means that the heritability of resistance, or if you want, the genetic basis of resistance, is close to 0. Which means that we should observe delays in the evolution of resistance if and only if farmers plant refuges close to their fields of Bt crop. So we know it's very likely that the refuge strategy that has been mandated by the US EPA has contributed in delaying the evolution of resistance in target pests, because we have demonstrated, many studies have demonstrated, that refuges are efficient in delaying the evolution of resistance. So what have been the consequences of pest resistance to Bt crops? Well, so far the consequences have been low because we only have 3 species that have evolved resistance. So in the fall armyworm that evolved resistance to Bt corn in Puerto Rico, the industry has withdrawn voluntarily Bt corn in Puerto Rico, which suggests that perhaps we are losing the potential benefits of Bt corn in Puerto Rico. In Helicoverpa, the evolution of resistance to the Bt toxins in Bt cotton may have increased the use of insecticides to control this pest in Bt cotton, but we don't have the data to assess this possibility. And in the western corn rootworm, the resistance is to Bt corn in some locations in the Midwest is recent, so we don't have data to assess the impact of such resistance. Now I'd like to talk about the issues that concern both Bt crops and herbicide-resistant crops, and those are gene flow between GE and non-GE crops, and gene flow between GE crops and weeds. So gene flow between GE and non-GE crops can occur for 3 reasons. First, it can be pollen mediated by wind, such as in corn. It can be pollen mediated by insects, such as in alfalfa and cotton. Or it can be seed mediated when farmers plant fields of a GE crop and a non-GE crop at close proximity in the same field. And this will occur for 3 reasons. Farmers may mix their seeds inadvertently at planting, so they mix seeds of GE, non-GE crops. Seeds left behind in a previous growing season could germinate in the wrong place. So for example, here, this is a Bt cotton field that germinated from seeds from the previous growing season in Arizona. So it's possible that this Bt cotton field would have germinated in a field of non-Bt cotton, which would have favored gene flow. And lastly, the seeds, the GE seeds and the non-GE seeds could be mixed after harvest. And if those seeds are used to plant the next crop, that would also favor gene flow. So based on the last 15 years, we know that gene flow between GE and non-GE crops is widespread and impossible to prevent completely. So what this means is that if you look at seed bags of non-GE corn, soybeans, and cotton today, you're likely to find the presence of GE seeds in those seed bags. So the presence of GE seeds will typically be less than 1% of the seeds, but it can be higher. So 2 potential negative impacts of gene flow between GE and non-GE crops is that it could accelerate the evolution of resistance to Bt crops if many Bt plants occurred in refuges. So far, this has not occurred in the United States or elsewhere. But the location most at risk for this would be developing countries where farmers recycle their non-GE seeds. We know that if you recycle your non-GE seeds, there's a greater risk of accumulation of transgenes, including transgenes that code for the production of Bt toxins. Another potential negative impact is that gene flow between GE and non-GE crops could limit the production of organic crops, and this would have indirect environmental impacts because organic agriculture is typically one of the most sustainable forms of agriculture. So what happens here is that organic crops are not supposed to contain GE traits in many countries. In other countries, the presence of GE traits will be tolerated. So for example, in Europe, you can have up to 0.9% of GE material in organic crops. So what would happen here is that if you have an organic producer that grows an organic crop in a region where you have a lot of gene flow, and if you have a lot of GE crops and the gene flow is high, it could be difficult for this organic producer to produce organic crops that meet those requirements for a low presence of GE material. So this is a question that has been brought in several publications, but when we wrote this report, we could not find data to rigorously assess this possibility. The second form of gene flow is between GE crops and weeds. And here we have 2 potential negative impacts. First, transgenes for herbicide resistance could be transferred to weeds in agricultural fields, and this would favor the evolution of resistance in those weeds and threaten the benefits of herbicide-resistant crops, as I talked about previously. Second, the transgenes for Bt traits or herbicide resistance could be transferred to weeds outside of agricultural fields, and they could spread in those weed populations if they confer consistent fitness advantages. So for example, there was a study done with sunflower where Bt sunflower was crossed to weedy sunflower. And the hybrids, weedy sunflower, were grown outside of agricultural fields. And what was found is that the production of seeds in sunflower, that weedy sunflower that was producing the Bt toxin, was significantly higher than the production of seeds in hybrid sunflower that did not produce the Bt toxin. And the reason for this is that insects were less able to feed on the seeds in the weedy sunflower with the Bt protein than in the other one. So this experiment suggests the possibility that Bt transgenes could consistently increase the fitness of weeds that would receive those transgenes outside of agricultural fields, and possibly those transgenes could spread. So the risk of gene flow between transgenic crops and weeds has been evaluated in the United States, and it's important to say that for the main crops, corn, soybeans, and cotton, this risk does not exist in the case of corn and soybeans, or is very low in the case of cotton. So the risk does not exist in corn and soybeans because there are no compatible weeds in the United States. So no possibility of transfer of transgenes. For cotton, there are compatible weeds, but they are only present in Hawaii and Florida. And the US EPA has restricted the use of Bt cotton in Hawaii and Florida to prevent such gene flow. So the risk there is probably very low. If you take crops like virus-resistant squash and herbicide-resistant sugar beet, the risk is there, but it's probably very localized. So for example, if you take sugar beet, it seems that the compatible weeds are only abundant in certain counties of California. So there, the risk of gene flow could be high, but not necessarily in other locations. So finally, if you look at crops like alfalfa and canola, you have feral plants there. That is, plants that have escaped agriculture and that grow outside of agricultural fields. So for these plants, if the feral plants are well established and produce seed year after year, you would probably look at a high risk of gene flow. So that would be the case for alfalfa. For canola, we know that the risk of gene flow is extensive, both to feral canola and to several species of mustard weeds. So here I'm showing you data that show you what I mean by extensive. This is a study that was done in North Dakota. And in North Dakota, feral canola that grows along road was monitored for the presence of transgenes that confer resistance to herbicides. So on this map here, the dots represent fields of canola. A lot of those fields are genetically engineered for resistance to either glyphosate or glyphosate. So the dots the circles in red here are feral populations where transgenes conferring resistance to glyphosate were found. In blue, you have populations where the transgenes conferring resistance to glyphosate were found. And in yellow, you have populations where both transgenes were found. So what happens here is that canola is harvested, put in trucks, and the trucks drive along the roads and the seed fall off the trucks. And the feral canola get established in those populations along roads. So clearly, here you have an example of an important gene flow to a weed population. So the next generation of GE crops will be engineered for several traits. I've produced a partial list here. They will be engineered for drought tolerance, tolerance to high salt content in soils, enhanced fertilizer utilization, carbon sequestration, efficient energy production, nutritional benefits, and production of industrial and pharmaceutical products. So for some of these traits, if the transgene is transferred to weeds outside of agricultural fields, it seems more likely that those transgenes would increase consistently the fitness of the weeds and therefore could spread more easily in those weed populations. This would be probably the case for drought tolerance and tolerance to high salt content, for example. Also, the last 15 years have shown that it's very difficult to prevent completely gene flow between GE crops and non-GE crops. So here, in the case of crops that produce industrial and pharmaceutical products, there is likely a risk of gene flow to crops that could potentially be grown for food of animals or humans. So basically, with those new traits and the new crops that will be modified, in the future, we're not only looking at measures to reduce gene flow, we're looking at measures, in some cases, to totally prevent gene flow between GE crops and weeds, and GE crops and sexually compatible crops. So when GE crops were commercialized in the United States, there were discussions about using molecular techniques named genetic use restriction technology to prevent gene flow between GE crops and non-GE crops, or GE crops and weeds. But the opposition of some stakeholder groups prevented the use of these technologies. So in the future, with all those new traits and new crops that will be transformed, it seems that perhaps we should revisit this idea, and perhaps we should use those genetic use restriction technology to prevent gene flow, at least in the case of the transgenic crops that will represent the higher risks. So on this, I'll stop here. I'd like to thank you for your attention, and I'll do my best to answer any questions that you could have. Thank you.